Alright, chapter 18, we'll be looking at blood. Alright, so again, we're going to be looking at blood hematology, what we have looked at in the past in 201 under the microscope. When we talk about blood cells, we're talking about red blood cells, and we're talking about white blood cells. White blood cells play a role in the body's immune system, while red blood cells carry nutrients and waste away. These are just some of the learning outcomes, the composition of plasma, the viscosity or thickness, osmolarity, the function of blood, and some of the basic general terms of blood and its production of it. Alright, circulatory system consists of the heart, blood vessels, and blood. Cardiovascular is the flow of the blood through the heart. Hematology is the study of blood. Function, it transports CO2, waste away, nutrients to the tissues, O2 to the tissues, regulates hormones, plays a role in the immune process, the white blood cells, B cells, T cells, T lymphocytes, B lymphocytes. It has lysozymes to destroy microorganisms, cancer cells. It neutralizes toxins, which are free radicals, in the form of antioxidants. It plays a role in blood clotting. It maintains our fluid and electrolyte balance, our pH, and body temperature. Adults produce about 4 to 6 liters of blood, depending upon male or female, and body mass. The matrix of the blood is clear, light yellow fluid, which is the plasma. The form elements, which is our white cells, platelets, and red blood cells. Red blood cells are called erythrocytes. White blood cells are called leukocytes. Platelets are called thrombocytes. And they all are developed from a common hemopoietic stem cell. Alright, erythrocytes, red blood cells, platelets, these are fragmented red blood cells specialized in the bone marrow for blood clotting. White blood cells are broken down into granular and agranular. The granulocytes are neutrophils, eosinophils, and basophils. Agranular are lymphocytes, which are B, would become antibodies, T, which becomes our T lymphocytes, and monocytes. And when monocytes mature, they become macrophages, which wander and circulate throughout the body. They can be fixed or they can be wandering. Alright, figure 18.1 shows you a picture what red blood cells look like. The monocytes, you can see red blood cells, biconcave, this shape. You see they are anucleated, no nucleus, so they stain pink or red. Where the white blood cells, you see all of them have a nucleus, such as our platelets, so they can stain different colors, and they are identified based on the size of their nuclei. When they talk about a CBC with differential, they're looking at the white blood cells, and differential, looking at the particular types of neutrophils, eosinophils, basophils, monocytes from that standpoint, and it lets us know what type of infection we have going on, acute, chronic, bacterial, and or viral. Alright, when you go get blood drawn, they send it to the lab, normally in the antibrachial groove there. The erythrocytes are heaviest. They settle first. They make up between 37 to 50% of the total volume. White blood cells about 1%. The plasma makes up 47 to 63%. It's a mixture of nutrients, electrocytes, waste, hormones, and our different body gases. A hematocrit, that's the centrifuge that separates the blood cells into its basic components. Plasma is the liquid portion remaining. It has no fibrinogen, which is the clotting mechanism. A lot of times when people say, well, we're going to get plasma, this is to help with raising the blood volume. So this has no clotting or solids in it. The categories are plasma proteins, the fibrinogen for clotting, globulin, antibodies, and albumin, which is abundant protein that helps to maintain fluid and electrolyte balance. Also, it regulates and influences our blood pressure based on the size and the number that are present. Nitrogen, urea is a nitrogen waste. When the nitrogen base is built up, that means that the kidneys are not functioning properly. Nutrients are glucose, vitamins, phospholipids, cholesterol. Some of the dissolved ingredients in blood, chemicals, oxygen, carbon dioxide, and nitrogen. Sodium potassium chloride are some of the electrolytes that we find. Now, all positive charged ions are cations, like sodium, potassium, magnesium, phosphorus. Chloride is a negative, it is an anion. Viscosity is the thickness of blood. Plasma is two times as viscous as water. But you want blood to have some viscosity to it, but if it gets too thick, then what that causes, it can cause blood to move at a very sluggish rate. That can cause it to adhere to the lining of the blood vessels and can cause blood clots. A clot that remains in a vessel is referred to as a thrombus. One that dislodges is an emboli, and those are the ones that can be dangerous and detrimental. Osmolarity, you remember we talked about this dissolved particles in blood. The thickness of it, if too high, blood absorbs too much water, raises the blood pressure. If it's too low, water stays in tissues, you get edema within the interstitial fluid. The optimal osmolarity helps to regulate adequate sodium, proteins, and blood cells. Hypoproteinemia, this is a deficiency of plasma proteins. When people are going through extreme starvation, kidney disease, liver disease, or people have about third-degree burns. The Kawasaki, children with severe protein deficiency, swollen abdomen, thin arms and legs. A lot of these have protein deficiency. Hemopoiesis is the production of red blood cells. Hemopoietic is the stem cell that produces and colonizes from the marrow, the spleen, the thalamus. But these are the cells that give rise to all the different types of blood cells based on the type of hormone that is going to influence it. So the adult produces about 400 billion platelets, about 100 to 2 billion red blood cells, and 10 billion white blood cells on a daily basis. The pluripotent stem cells, the hemocytoblasts, blast means immature, or your hemopoietic stem cells. Colonizing forming units, these are specialized stem cells that produce a particular class of blood elements. Myeloid hemopoiesis, blood formation in bone marrow. Lymphoid, these are blood formation in the lymphatic organs. Now remember, the lymphatic organs, they play a role in filtration impurities in the body. Alright, let's look at erythrocytes. Alright, two principal functions. They carry oxygen from the lungs to the tissues. They pick up CO2 at the tissue site, bring them back to the lungs to be excreted. If we don't have enough red blood cells, this causes death in minutes due to a lack of oxygen to the tissues. And this can, when people have strokes or when they're drowning, and when the brain goes without oxygen for too long a time period. Again, remember, red blood cells are biconcave disc shape. They have no mitochondria. They have no nucleus, no DNA. They have a lifespan between 100 and 120 days. Their main purpose is to carry O2 and nutrients to the tissue, pick up CO2 from the tissues, and carry them to the lungs to be excreted out. Formation, again, blood types determined by the glycoproteins and glycolipids on their surface. They have spectrin and actin that gives them mobility and resilience to go from large blood vessels to very tiny ones, or the capillaries at tissue exchange level. Gas transport, hemoglobin has 4 binding molecules, 2 alpha units, 2 beta units, so they can pick up O2 and carry CO2. You got 280 million hemoglobin molecules on one red blood cells. Carbonic anhydrase is a enzyme that plays an important role in regulating the acid-base balance in the body. And when you get to nursing, this is going to play an important role in fluid and electrolytes. All right. Alright, so again, hemoglobin has 4 binding units. Now, when we talk about sickle cell anemia, there's a discrepancy in the genetic code, and then the red blood cell, instead of being biconcave, is going to become sickle shaped. So fetal hemoglobin contains 2 alpha and 2 gamma chains, where adult hemoglobin has 2 alpha and 2 beta chains. So heme is the iron component of the globulin protein molecules. So a hemoglobin is made up of proteins and iron. Alright, hematocrit, this is packed blood cell, 42 to 52% in males, 37 to 48% in females, based on the body mass per man. Hemoglobin, 13 to 18, 12 to 16 in females. Now remember, females have a menstrual cycle that affects their blood, total blood concentration. The RBC count is 4.6 to 6.2 in males, and about 4.2 to 5.4 in females. So we can see vagus are low in women. Androgen plays a role in the red blood pressure production, but menstrual loss and also the hematocrit is inversely proportional to body fat mass. Alright, this is a picture of figure 18.6 of erythrocytes. Start out with a pluripotent stem cell. Then you got a colony forming unit that looks at the erythrocyte. And look at, as we go from left to right, how the nucleus gets smaller and smaller. Then the precursor cell, you get erythroblast. Blast means immature. Reticulocyte, it has fragmented, and erythrocytes a mature red blood cell. So development is about 3 to 5 days. And you see the size goes from a large cell to a very small one where the nucleus is completely lost. Alright, again, just a continuation. The first committed cell you're going to be looking at is the colony forming unit. Now once that cell is formed, then it's going to automatically go on to a mature erythrocyte. Erythroblast multiply and synthesizes hemoglobin, and then from there the nucleus is excreted out. Then we have a mature erythrocyte. Okay, so it makes up about 0.5 to 1.5 of the circulating red blood cells in the body are reticulocytes, which are immature before they mature to the erythrocyte. Alright, iron metabolism, you see the liver, you see the stomach. Now remember, hemoglobin is made up of iron and globin. Heme is the iron component. So again, the transferrin is distributed to the other organs where iron is used to make hemoglobin or myoglobin in muscles. Iron can bind to a protein, apoferritin, and there it can be stored as ferritin. Then let's look at iron. It goes to the digestive tract. You say iron 3. Then iron 3, you lose a plus component. It goes to iron 2. There it binds to gastroferritin in the stomach. Then it goes into the small intestine. The gastroferritin transports iron, and it's going to be released so for absorption. There it's going to go into the liver. There the ferritin is there, and the apoferritin, they bind. So there in the liver, some of the transferrin is released for storage. The other binds apoferritin, where it's going to be transported to the other organs in the form of F2. Here, iron is a major nutrient that we need. You cannot make red blood cells without iron. So 0.9 in men to 1.7 in females has a low absorption rate is required based on the consumption per day in milligrams. Alright, so this just shows you that the stomach acid converts iron 3 to iron 2. It's going to be going to the small intestine. There it's going to be absorbed and binds to transferrin to the bone marrow and the liver and to other body parts there. The bone marrow forms hemoglobin, muscles myoglobin. You got the cytochromes and the mitochondria that reduces it down. The liver has the apoferritin that can bind to create ferritin for storage there as is needed. B12 and folic acid is very important in red blood cell production. When you talk about anemia, you can have B12 deficiency and folic acid deficiency. Copper and vitamin C are important in the synthesizing and making of hemoglobin. Copper is moved around in the blood by the alpha globulin called ceruloplasmin. Here, hypoxia is a decrease in O2 content based on the drop of the red blood cells. When this happens, it stimulates negative feedback. It causes the kidneys any low oxygen content to produce the hormone erythropoietin. This acts on the bone marrow to produce and increase red blood cell production 3 to 4 times more per day. So it's stimulated by low O2 levels, high altitude, increased exercise, or emphysema. That emphysema is COPD, which causes a decrease in the body to bring in oxygen. Alright, erythrocyte death and disposal, hemolysis, the breakdown of these blood cells. Hemoglobin is broken down by macrophages. It's a mature monocyte. It's digested. Globin hydride is broken down back into amino acids. Iron is removed in the form of heme. It goes into our bile system. Bilirubin is green. Bilirubin is yellow. It can be released back into the blood plasma in the kidneys to form urine and make it yellow. Gallbladder released into the small intestine. Bacteria can convert it to urobilinogen that gives our fecal matter its brown color and its odor. Alright, this is just a schematic that goes on after between 120 days. Red blood cells is broken down. The amino acids and iron and stuff are released. The erythropoiesis process stimulates the red blood cell to make more, circulates throughout the circulatory system for about 100 to 120 days. Expired erythrocytes break up in the liver and spleen. The fragments are reutilized and reabsorbed. Some is excreted. The other can be retransmitted to be used to make new red blood cells. Polycythemia is a disorder of the red blood cells in terms of cancer. Primary polycythemia, again, Vera, is a situation when cancer of the erythropoietic cells, we produce excessive amounts of red blood cells, all right, like 11 million, very thick, very thick viscosity, hematocrit up to 80. From dehydration, emphysema, high altitude, RBC count can go up to 8 million. This is a secondary type. Increase in the blood volume, pressure, viscosity causes too much demand on the heart, and then that can result in embolism, heart failure, and massive strokes. Again, different types of anemia, inadequate erythropoiesis to stimulate red blood cell production. Iron deficiency, autoimmune disease can be associated with pernicious anemia, where we get inadequate B12 absorption. Hypoplastic, slowing of the erythropoiesis process. Aplastic, you don't have the erythropoiesis being produced at all. Hemorrhagic bleeding, and hemolytic is destruction of our red blood cells. Tissue hypoxia is another type in which that we look at patients' lethargy, shortness of breath, can cause necrosis or hardening of brain, heart, and lung kidney. Reduced osmolarity due to tissue edema and viscosity. So heart races and pressure drops, and you can go to cardiac failure. This is just a picture of sickle cell anemia, where the blood cells are no longer biconcave disc, but they are not biconcave but S-shaped, and they become hard and rigid. They can cause pain as they move through the blood vessels, kidney pain, heart pain, joint pain from this standpoint. One thing about it, if you have sickle cell trait or disease, you're resistant to malaria if you are in those areas that has an epidemic outbreak of malaria. Alright, again, blood types we're going to look at. Again, we look at the red blood cells. We have A, B, O, AB, and then we have the component AB positive, AB negative based on the rhesus component. Blood cells have antigens and antibodies attached to them, determining blood who can and who cannot receive blood. Antigens, these are things that elicit an immune response. They are unique genetically to every individual. They are viewed as foreign if given to the wrong person. Agglutinogens, this occurs when antigens on the surface of the red blood cells that are basic for blood typing. So agglutinogens are the things that we look at in terms of agglutinating or causing the clumping of red blood cells if the wrong blood type has been given. Antibodies, these are the ones that fight off the infection, part of the immune response cells. They form what are called anti-antibody complex or agglutins. This occurs when the blood type is given incorrectly. So the molecules combine the antigens and cause clumping of the red blood cells, or what we call agglutination. Alright, agglutinogens, red blood cells, called antigen A and B. Alright, they're determined by the glycolipid configuration on the cell. So let's look. Galactose, fructose, fructose, and intercellular galactosamine. These are highlighted on the right in figure 8.12. Antibodies are called agglutins. They are found in the plasma, anti-A and anti-B, where the agglutinogens, these are the antigens in terms of A and B. Alright, so a person blood type A has A antigens. A blood type B has B antigens. AB has both A and B antigens on them. Person O has neither antigen on them. So again, A antigen. Alright. B antigens. Okay. A, B, both, and blood type O, neither antigens on them. Okay. Alright, so let's look at AB blood type. You see A, you see anti-A, then anti-B. You see type B, where it reacts with A. AB, you can see what happens in O that has neither. Alright, so again, the agglutinogens, anti-A and anti-B, about the second month after birth. Alright, the concentration is at its maximum adult rate about age 10. Antibody A or B or both or neither are found in the plasma. Alright, these are antibodies that interact with the agglutinogens and they cause the clumping of the red blood cells. Agglutination, each of the antibodies can attach to several forward antigens, so this causes a transfusion reaction. So the hemoglobin blocks the kidney tubules and causes acute renal failure. Agglutinated red blood cells block the smaller vessels called hemolysis that release the hemoglobin over a couple of hours or days. Alright, so this is a blood from type A donor. Alright, you can see the blood. They got type B, anti-A recipient. They donate the blood. Agglutination of the red blood cells begin to take place because type B has antigens A on it. So therefore, if it's a type A donor that goes to a B person, that's going to cause agglutination to take place. Charles Drew was a scientist that came up with blood banking and blood plasma for blood transfusion. And ironically, he was Afro-American, and he was in a car accident, and he ended up being in an automobile accident, and he ended up dying from lack of a blood transfusion because he could not be admitted to a white hospital. And by the time they got him to an Afro-American facility, he had expired. Alright, so our AB group in type O, most common type, donor plasma have both antibodies against recipients, or RBs, anti-A and anti-B. So they may give packed blood cells. Universal recipient, they lack the antibodies so they can receive. Alright, so O can donate to antibodies, but AB can receive from antibodies. Rhesus, D, most reactive of the RH factors. It tends the agglutinogens on RB red blood cells. Anti-D agglutinins form the RH negative individual and RH positive. This occurs with what we call erythroblastosis fetalis or hemolytic diseases in newborns. If the mother is RH negative and she marries the father who's RH positive, and if they have a baby that's RH positive, the anti-D antibodies can cross the placenta, and this would cause a reaction in the mother. The first pregnancy would be okay, but her body automatically produces antibodies against that baby. So if she gets pregnant again, that can elicit an immune response in her system to view that fetus as abnormal if the baby is RH positive. So therefore, her body will automatically produce antibodies that are there that will become active again and try to kill the baby. So what we do is we give RhoGAM to the pregnant women that we know that the father is Rh positive and the baby is probably Rh positive, and this has to prevent that from taking place. Alright, figure 1816, it just shows you the Rh antibodies that can attack the fetal blood causing toxic brain syndrome or severe anemia if it's not treated early on, or this is what we call hemolytic diseases of the newborn. Alright, leukocytes. These are our white blood cells. They manufacture between 5 to 10,000. They do have a nucleus. They have different sizes. All white blood cells have lysosomes, aerophilic, non-specific, but they have the ability to digest and destroy invading particular type of organisms. Granulocytes, these are specific, where we got the eosinophils, the neutrophils, and the basophils that carry out specific defense mechanisms in our body. Alright, neutrophils make up 60 to 7%, visible in the cytoplasm, the 3 to 5 largest of the nucleus. Eosinophils, again, mainly for allergic type of reactions. They are bilobed nucleus, make up 2 to 4%. Basophils for our reaction that increases the diameter of blood vessels. A-granular lymphocytes, these are our B and T lymphocytes, make up 25 to 33%. They can be dark violet nucleus, scanty, ovoid around. Monocytes, they're immature, but when they mature, they become what we call macrophages. Okay, so neutrophils are aggressively antibacterial. So increasing their numbers, it knows we have an acute bacterial infection. Eosinophils are allergic type of reactions, parasitic worm infections, things that interfere with the central nervous system. So they release enzymes to destroy large parasites like worms or cysts that we may get in undercooked pork or chicken. Basophils, we see these with chicken pox, sinusitis, diabetes. These are what we call vasodilators. They allow to increase flow of blood into injured areas. Heparin is an anticoagulant, so they also increase the mobility of the white blood cells to the area compromised. Figure table 18.6 shows you a granulocyte. Agranulocytes are lymphocytes, B and T. They present antigens to activate other immune responses, be it the antibodies that work along with the T cells. So they have to fight off viral, cancerous, and any type of foreign entity that may have entered the body. Monocytes, when they mature, they become macrophages. These are what we call the antigen presenting cells. Once they find an antigen, they will present them to the other type of cells, be it viral, inflammatory, in order to elicit an immune response to destroy that agent, such as a pathogen. And a pathogen is any organism that can elicit an immune response. Agranulocytes, you can see here, these are lymphocytes, our monocytes. Hemopoietic stem cells, myeloblasts, they make the neutrophils, eosinophils, basophils. Monoblasts make monocytes, and our lymphoblasts, they can make our T lymphocytes that develop and mature in the thalamus. Those that mature in the bone marrow or the plasma cells become the antibodies. Alright, again, the white blood cells, they do not stay, they circulate throughout the system and the tissues to fight any type of invaded organism. In 20 hours, once they leave, they transform to macrophages. Macrophages can be fixed in the lungs, they're called alveolar macrophages, or in the liver, Kupffer macrophages, or they can be wandering. They have the ability to migrate and move throughout the body. So they provide us with our long-term immunity or immune system. They can last for years. Alright, learn this diagram. This shows you the pluripotent stem cell, and based on the committed cell and what they're going to develop into. Like the eosinophils, they become the eosinophils through the eosinophilic myoblast to the eosinophilic myelocyte to the eosinophilic myelocyte to eosinophil. Same thing with the neutrophils. So again, our lymphocytes, they can be B or T. The B cells mature in the plasma, become antibodies. The T in the thymus to become different types of T cells, helper, suppressor, killer, or delayed hypersensitivity T cells. Okay, so again, as I stated, make sure we look at this chart to look at the different blood cell production. Alright, leukopenia is a low white blood cell count below 5,000. Normally radiation, poisons, infectious. People are at a higher risk of infection. Leukocytosis is elevated. It can be associated with different type of infections, allergic type reactions. Leukemia is cancer of the white blood cells. Myeloid is uncontrolled granulocyte production. Lymphoid, lymphocyte or monocyte production. Acute, progressive rapid death within months. Chronic, normal survival time, undetected, but for 3 years survival rate. Normally with leukemic leukocyte disorders, people are prone to opportunistic infection. An opportunistic infection is one that cannot cause you to be sick if you're healthy, but in those who have a compromised immune system. Alright, this is just a histological view of what a normal red blood cell slide looks like with the white blood cells and the red blood cells. Alright, complete blood count, you're looking at the differential white blood cell count, hemoglobin, hematocrit, reticulocyte count, as well as platelets. Alright, hemostasis is stabilizing normal red blood cell. So hemostasis is stopping bleeding or excessive bleeding. Vascular spasm is one, platelet plug, and blood clotting. The platelets is the essential component and blood clotting mechanism that we're going to touch right now. Alright, platelets again, they're megakaryocytes, very large, they fragment. The normal count is 130 to 400,000. They're amoeboid-like, and they have phagocytosis type of capabilities. Alright, again, platelet plugs, seal small breaks, procoagulants. These are your pre-clotting factors, initiate the formation of clot dissolving enzymes. Once a clot is formed, you want to make sure you got an enzyme embedded in that clot to dissolve it to keep it from forming a thrombus, and that's the internal clot. Chemically, through chemotaxis, they can attract neutrophils and monocytes in order at the site of inflammation to help regulate it and to control it. Alright, these are what the platelets look like, figure 18.28 and B. Alright, a thrombopoiesis, the stem cell that produces the platelet, a megakaryoblast. When you keep duplicating the DNA without dividing, it gets very large. Then megakaryocytosis in the bone marrow adjusts through the blood sinusoid. Then they begin to kind of fragment to protrude out. They normally circulate 5 to 6 days, and then they're stored in the spleen. Right. This is a platelet plug. You got the vascular spasm where you get intermittent contraction. Each time it contracts, it closes, but when it releases, it opens again. And then the last one is the clot. Alright, hemostasis, again, intermittent protection. Pain receptors, smooth muscle injury can cause it. Serotonin, which is a vasoconstrictor. Effects provides time for the clot pathway to take place. Alright, prostacyclins, a platelet repellent. Broken vessels allows the collagen to be exposed. You get pseudopod sticky-like to the damaged vessel wall. It draws in the plug. Degranulation releases other substances such as serotonin that act as a constrictor. Now, we get some other components like thromboxanate 2, eicosanoids, your platelet aggregation factors, degranulating, and vasoconstriction. This is a positive feedback cycle to initiate this activated until the break of the vessel has been walled off and sealed. Again, coagulation, clotting, you got fibrinogen, is a insoluble fibrin thread, procoagulants, clotting factors, again, activated cascade of events, either intrinsic or extrinsic. Extrinsic, outside, released by damaged tissue. Intrinsic, within the endothelial lining. Learn figure 18.22. It shows you both extrinsic, where the damaged vessel occurs, thromboblastin, factor 3 is released, then 7, calcium comes in, you activate factor 10, then from there you get the liver, you got prothrombin, factor 10 activates the prothrombin activator, you convert prothrombin to thrombin, then that's the enzyme that takes and converts fibrinogen and fibrin and forms the clot. Intrinsically, you get the same result, but you start off with the platelets releasing factor 12, then they activate 11, then 9, then you got your calcium, your PF3 factor. Now, when we look at hemophiliac, you got factors A, B, C, or the clotting factors are missing, and that causes the free bleeding to take place. Okay, if you look at figure or slide 79, calcium is required in both pathways. In extrinsic, you're looking at the inner lining is damaged, factor 3, then 7, 5, and 10. You got fewer steps as compared to the intrinsic Hagman factor 12, and it initiates a sequence of internal events that eventually lead to the fibrin clot. Alright, this is just our cascade and clotting figure 18.23. Activation of 10 leads to the prothrombin activator that then converts prothrombin to thrombin. Then thrombin is the activator that converts fibrinogen to fibrin. Factor 12 cross-links fiber polymer strands. So this continues until the bleeding time has ceased, knowing about 1 millimeter deep depth of the incision. Clot retraction occurs within 30 minutes. Platelet-derived growth factors secreted, mitotic stimulation for fibroblasts, and then we see repair of the damaged vessel. Kallikrein enzymes converts plasminogen to plasmin, and then this is a fibrin-dissolving enzyme that breaks the clot down to cut down from an internal emboli. Alright, this is just a schematic of our blood clots that's taking place. Alright, then we look at platelet repulsion. Again, do not adhere to the inner lining. Thrombin dilution, heart slowing and shock can result in a clot to form, or your natural heparin anticoagulants and antithrombin. Antithrombin is formed from the liver. Alright, clotting disorders, hemophilia. Hemophilia A is missing factor 8, 83% of the cases. B is 9, 15%, and 11. And autosomal is hemophiliac C. Alright, again, clotting disorders, transfusion of plasma or purified clotting factors. Factor 8 that can be produced by transgenic bacteria. Hematomas is a blood clot that forms under the tissue, under the nails, secondary to trauma. Thrombosis is an internal clot in the vein itself. Pulmonary embolism, one that has broken free, travels to the lung. Infarction is blockage due to a clot in the major arteries of the heart that can cause death, or we call it thromboembolism, that's traveling in the blood. Preventing clots, we have vitamin K that is produced, Coumadin, warfarin. These are those things that act as anticoagulants. Aspirin suppresses thromboxane 2. Again, aspirin is one of those things that helps to thin blood, to help cut down on clotting. Alright, again, streptokinase enzymes made by streptococcus bacteria help to digest almost any protein. Tissue plasminogen activator works faster and made of transgenic bacteria. And hematin is produced by Amazon leaks. So this concludes chapter 18.